Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. A gene that is inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Certain viruses are often used as vectors because they can deliver the new gene by infecting the cell. The viruses are modified so that they can't cause a disease when used in people. Some types of viruses, such as retroviruses, integrate their genetic material, including the new gene, into a chromosome in the human cell. The vector can be injected by IV directly into the specific tissue in the body, where it is taken up by their individual cells, or alternatively, a sample of the patient cell can be removed and then exposed to the vector in a laboratory setting. The cells containing the vector are then returned to the patient, and if the treatment is successful, the new gene delivered by the vector will make a functioning protein. Researchers must overcome many technological challenges before gene therapy will be a practical approach to treating disease. For example, scientists must find better ways to deliver genes and target them to the particular cells. They must also ensure that new genes are precisely controlled by the body. Brian Mordew, a 44-year-old patient with Hunter's syndrome, is the first patient in which scientists have actually tried to edit a gene inside a person's body in an effort to cure disease. His doctors described the process by telling him that they will cut his DNA, open it up, insert a gene, and stitch it back up. So basically, it's invisible mending. So it becomes part of one's DNA and therefore is there for the rest of your life. A major worry is that inserting a new gene might have unforeseen effects on other genes. This did happen years ago when researchers used gene therapy to cure some cases of the immune system disorder called bubble boy disease. Several patients later developed leukemia because the new gene inserted into a place in the native DNA where it unintentionally activated a cancer gene. This begs the question, what other harm is it causing? According to Hank Greeley, a Stanford University bioethicist, the advantages with the new development of gene editing is that you can put the gene in wherever you want it to go. So research has progressed to experimenting with ways to control the outcome of the inserted altered DNA strands in other parts of the body as well. The only problem is, will this work, and who will be the next test subject?
According to Gullins, a bioethicist, it is known that viruses are very good at inserting themselves and modifying human gene code, and therefore, in order for a virus to survive, it has to manipulate and replicate the DNA sequence, causing a genetic mutation. Gene therapy will therefore entail the identification and isolation of a single faulty gene and replace it with a properly functioning alternative. This replaced gene will soon take over and thrive by changing the entire genetic code. As far as clinical application is concerned, gene therapy holds promise for treating a wide variety of diseases, including cancers, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. According to Rochelle, another physician, many physicians are currently predicting through research that in 20 years' time, genetic therapy will change the practice from a treatment-based to a preventative-based practice. The implications of gene therapy related to the conditions covered by physiotherapists have the potential to be life-changing for the patients.
The ethical concerns relating to gene therapy in humans. Number 1, ethics versus religion. When it comes to human modification, will the church remain in submission to the state? Number 2, ethically, where do we draw the line? And finally, the ethics concerned with human test subjects.
Recommendations for the future physiotherapy practice with regards to gene therapy.